Hi everyone from the Upside Down. I'm going to make this a theme now. Today we're going to do chapter 20, which is Cancer. Why do my eyebrows look funny? Okay, it's fine. Alright, so Cancer, the question of the day down here is, will we ever be able to prevent cancer from forming? And I'll tell you right now, the answer is no, and we're going to find out why. And if I bring up my mom a lot, I apologize. This cancer stuff hits close to home, as I'm sure it does for a lot of you. I lost my mom to brain cancer, and then a few years later, my paternal grandma also died of brain cancer. So my OCD brain is like, that's how you're going out. It's in your genes. But based on what we learned here, that hopefully will not be the case. But yeah, my dad also had cancer. He had thyroid cancer. My grandpa passed away from lung cancer. And I'm sure you all have similar stories. But I will say, I took a class called the Biology of Cancer when I was in college, and it was somewhat helpful to know that when the doctors were talking, I kind of knew what they were talking about and my understanding of physiology as well. So this is a good thing to understand, especially if you are pre-med or anything of that sort. So let's get going. I do want to go over the pre-lecture assignment together because there are some terminology words that you will need to know. So cancer is a type of disease characterized by uncontrolled cell growth. So when we're talking about cancer, it kind of wants to do three things. It wants to grow uncontrollably. Wait, I don't know how to spell uncontrollably. Is that a word? That looks funny. Okay. It wants to be abnormal. And it wants to be immortal. To live forever. Alright, characteristics of cancer cells. And the first one is lacking differentiation. So cancer cells are non-specialized. So typically the cells in your body have differentiation. They have a different function and therefore they have a different structure. They're differentiated, but cancer cells are non-specialized and they do not contribute to the functioning of a body part. Another characteristic is they have abnormal nuclei. So remember, the nucleus of the cell is where all the DNA is contained, and the nuclei of cancer cells are enlarged, and you can actually see this under a microscope, and may contain an abnormal number of chromosomes. So looking at them under a microscope, they look all kinds of wacky. They fail to undergo apoptosis. So remember, apoptosis is that programmed cell death, and I remember the pop part where the cell just kind of gone. It's dead. Cancer cells fail to do this. No programmed cell death. Unlimited potential to replicate. So cancer cells are immortal. And if you read in the book, it talked about HeLa cells. I don't know if you read it in the text, but there's a book, I think it's called The Immortal Life of Henrietta Lacks. And it talks about some of her cancer cells that were taken, her cervical cancer cells, and this was in, I forget what year, but however many decades later, those cells are still dividing, and you can find cultures of them all over the world in labs. So, they are immortal. They will keep dividing an unlimited number of times, which is one of the things they want to do. And the chromosomes are capped with something called telomeres. That get shorter after each cycle, but in cancer cells, these are constantly rebuilt. So this is like a fun fact that everyone says. If you have a shoelace, that little tip at the end is it an aglet? I think that's what it's called. But think of your chromosomes as having little aglets at the end, and it's to protect them from fraying. And in normal cells, they kind of break down each time they divide, but in cancer cells, they're constantly rebuilt. So if here's our little chromosome here, on the end, you have these little telomeres to keep the DNA and the chromosome from unraveling. Cancer cells constantly rebuild those so the DNA doesn't break down, and it can keep dividing and being immortal. Another thing is that they lack contact inhibition. So normal cells, when they are dividing, if they come in contact with another cell, they're like, oh, we've grown enough. There's enough room here. We're going to stop dividing. And cancer cells don't do that. Instead, they pile on top of one another, forming a tumor. So that's what we see in this picture over here. Is normally the cells, when they start getting squished like this, would stop dividing, but you can see they continue dividing, they pile on top of each other, and they actually start infiltrating the tissue around them. And that's what we call a tumor. And these cancer cells undergo something called angiogenesis. So angio is referring to like blood vessels. And then remember, Genesis is formation of, kind of like Genesis in the Bible is about the formation of the earth. And so they can actually create their own blood vessels around them to get nutrients and oxygen and all those things from the body. So a tumor must have a well-developed capillary network to bring it nutrients and oxygen. So tumors form new blood vessels through a process called angiogenesis. And that's also what we're seeing in this picture over here. So if this were like a cross-section of a certain type of tissue, here's a lymphatic vessel, here's a blood vessel, and as it infiltrates and invades the tissue around it, it can actually start to utilize those vessels. And that's where metastasis comes in. So if you've heard of metastatic cancer, it's cancer that has spread. And so they can use the vessels of the body here to jump off and hitch a ride to somewhere else in the body and start growing on another tissue and form tumors other places. So when cancer cells invade blood vessels, they migrate to other parts of the body, and metastasis is the formation of a tumor far away from the primary tumor. All right, so from here, the takeaways from this page, I would say, are knowing the things that cancer cells should or want to be able to do, and that's grow uncontrollably, be abnormal, be weird, and never die. The vocab I would take away from here are the telomeres, okay, those little caps on the end of the chromosomes that cancer cells do not, they keep rebuilding rather than them breaking down like normal cells. And then I would know what angiogenesis and metastasis means. Alright, so on to the first page of our notes. We're going to talk about two different things, cyclins versus checkpoint proteins. So cyclins are proteins, and before I go any further, remember that proteins are coded for by your DNA. So DNA codes for proteins, which basically make up you, and these proteins have these specific functions. So these cyclins are proteins that trigger the next phase of the cell cycle, so think of them as being an usher, if you know what an usher does, to the cell through the cell cycle. So think of like going into an airport. Airports are a nightmare for me. I hate large crowds of people. I have no sense of direction, and I get like overstimulated by a lot of lights, noise, and people. So I hate airports. But think of like an airport, the ticket agent is you're going to walk in, you're going to figure out where you're going, like what flight company, airline company you're on or whatever, and they're going to usher you to the next area. They're going to imagine they're being super nice and you're feeling lost, and they're actually going to hold your hand and walk you to the next area. And security is the next checkpoint before you move on to the next area. And if there are any problems, if you have a gun in your bag, or if you have a knife or something abnormal that shouldn't be there, security will fix it before you can move on. So these cyclins, think of them as like the ushers helping you through the airport. They're helping cells through the cell cycle. So a type of cyclin is a proto-oncogene. So proto means before, and onco, anytime you see onco, that means cancer. That's why if you have cancer, you see an oncologist, someone who studies cancer. And these proto-oncogenes are genes that code for cyclins. Alright, so a proto-oncogene is normal, and it codes for these cyclins that help usher the cell through the cell cycle. But what can happen is these proto-oncogenes can become mutated So remember, a mutation is a change in the DNA, and they can become mutated to something called oncogenes. So now we've taken the proto off, and now it's an oncogene, so it's a gene for cancer. And just a reminder over here, I think we're talking about inheritance next. I don't remember if we talked about mutations already, but it's a change in DNA sequence where there are mistakes in the DNA. There can be deletions, part of the DNA gets removed, or insertions, you have extra DNA that's added that shouldn't be there, and so forth. Okay, checkpoint proteins. Remember, as you go through the cell cycle, you have these checkpoints that make sure that the cell is okay, it's good, it's got everything it needs, the DNA is replicated correctly, so it can move on to the next part of the cell cycle. So these checkpoint proteins, if a checkpoint fails, checkpoint proteins inhibit or stop the next cyclin to make repairs. So they're going to stop it, say, hey, what's wrong here? We got to fix it. And some checkpoint proteins are tumor suppressor genes. Genes that code for checkpoint proteins. And they can also become mutated. So if these tumor suppressor genes become mutated, then they're not going to inhibit the cell cycle if there are mistakes. And so this can allow cancer cells to continue to divide. So an example over here, and we'll label these in a second. G1, sorry, my G's are funny. Cyclin ushers a cell through G1, the G1 checkpoint. There we go, that's better. At the end of G1, the G1 checkpoint protein sees damage, the DNA, so what it will do is inhibit the S phase cyclin until it can repair the DNA. Oh, I just realized I'm off camera. I'll slide it over in a minute. Alright, so here's an example of how these things work. So G1 cyclin ushers a cell through G1. Got G1 right here. And at the end of G1, got the G1 stop checkpoint. If that protein sees damage in the DNA, it inhibits it from going into the next phase, the S phase, by stopping that cyclin from ushering it to the next phase until it can repair the DNA. So let's label our cyclins here on our cell cycle. So remember, those are proteins that trigger the next phase, usher the cell through the cell cycle. And it's pretty easy to remember because they're just named after the phase that they are associated with. So the cyclin that works here is called S-cyclin. The cyclin that ushers the cell through here is, surprise, G2 cyclin. The cyclin that ushers the cell through here is called M-cyclin. And the cyclin that ushers the cell through the cell cycle here is called the 782496 cyclin. I'm just kidding, it's called the G1 cyclin. All right, so it should be pretty easy to recognize which cyclin goes in. Sorry, I'm not that funny. Okay, so proto-oncogenes, remember those are the unmutated kinds, accelerate the cell through the cycle, and tumor suppressor genes skip repairs and allow cell through damage. That's worded funny. Hang on, let's go down to this pause and think part. So pause and think of what proto-oncogenes and tumor suppressor genes do in their mutated form. So hint, remember that cancer cells are abnormal and grow fast. So again, and this is where it gets a little hairy and confusing, proto-oncogenes, these are normal. It's when they become mutated that they become oncogenes and don't do their job. Tumor suppressor genes are normal, but when they become mutated, they stop doing their job properly. So if we pause and think here about what the mutated cells do, that's what I was just reading to you. So when proto-oncogenes are mutated, they accelerate the cell through the cell cycle. All right, so you got those ushers, and they're like, let's go, let's divide. And that leads to uncontrolled cell division and therefore cancer. And if tumor suppressor genes are damaged, they skip repairs, or if they're mutated, and allow the cell through damage. So again, another thing that cells want to do is they want to be abnormal. Okay, and I know that gets a little confusing. These are normal, and when they're mutated, it both says here can become mutated, that's when you start to see the problems. The cell's going to divide uncontrollably. It's going to pass through these checkpoints even though it's abnormal, and that's where we get these tumors. So a little bit more about proto-oncogenes. They code for cyclins, and they are dominant genes. And what that means, we're going to get into this in our genetics coming up next, but you only need, remember for all genes, you get a copy from mom and a copy from dad, and you only need one mutated copy for cancer to occur. That's an O. So you've got the chromosome from mom, the chromosome from dad. Let's say there's the gene for proto-oncogenes located right here on the chromosomes. And the one that you got from dad, we'll say, has a mutation. So that proto-oncogene is not going to work. And that's all it takes for you to have cancer. This is going to be normal for mom. But since it's a dominant gene, that mutated gene is going to dominate over that normal gene, and you only need one copy for cancer to occur. So some examples of proto-oncogenes, and these I would know for the exam, is called HER2. And this is a breast cancer gene. So not to be weird, but the way that I remember it is her breasts, okay? So it's associated, it's the breast cancer gene. And these genes code for estrogen receptors on mammary cells that trigger mitosis. And 30% of breast cancers contain many copies of this gene. So females have these mammary cells in their breasts, and they have these little estrogen receptors. I have no idea if this is how they are shaped. But when estrogen comes and binds to those, it causes the cells to divide. It triggers mitosis. And so this breast cancer gene is a mutation that codes for a whole bunch of these estrogen receptors to be on these cells. And so what's going to happen is more estrogen is able to bind to these receptors, and guess what? You're going to have uncontrolled cell division. One of the things that cancer cells like to do, divide uncontrollably. Alright, so what to remember here is HER2 is a breast cancer gene, and it codes for those estrogen receptors on the mammary cells in the breasts that trigger mitosis, cell division. I'm going to star this. That's something you should know. And also know that it's a proto-oncogene. Another proto-oncogene is RAS. And this gene activates cyclins as needed. So it kind of controls the activation of those cyclins that help usher the cell through the cell cycle. So pause and think of what HER2 and RAS genes do in their mutated form. So when you have a mutated HER2, it inserts, that's kind of what I showed in the picture, even more receptors. So that mammary cells divide even in low estrogen. And then when you have a RAS mutation, causes the cyclins to never shut off. So you get continuous cell division. I'm going to leave this here for a second in case you need to get caught up. So you may have heard of a diagnosis of HER2 positive. And this means that you have more of these receptors than normal. Okay, so HER2 in its normal form codes for these estrogen receptors. Just a few of them. But when it's mutated, it inserts even more of those receptors, so more estrogen can bind, and that will trigger mitosis. The RAS gene, in its normal form, activates cyclins as needed to usher the cell through the cell cycle, but in its mutated form, the cyclins are just always there, and so again, they're just like, get through, go, 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 get through the cell cycle, we gotta go, and the cell just keeps rapidly dividing. So there's a paper for this, and somehow I don't have it printed off, but what I would do is after this, and I think I'll have to check Canvas, I think it's a separate link to print off, it's like a cancer table, and it has proto-oncogenes, and I think the proto-oncogenes are, can't remember if they're on the top or bottom, and then cyclins, and so it has them divided into two categories, and then it lists the genes, the examples, and then it has a column where you write what it does as its normal duty, and then what it does when it's mutated. And I would recommend going through and trying to fill that out. That would be a great way to study. And then that table is also something that you should know for the exam. This is one you should know as well. Okay, so tumor suppressor genes, these are recessive genes. And what that means is you need both copies to be mutated for cancer. Right, so if we draw the picture of the chromosomes here, one from mom, one from dad, let's say these tumor suppressor genes are located right here. If you got a mutation from dad, but not from mom, since it's recessive, you wouldn't get cancer from that. In order to get cancer, you have to have both of those genes, one from mom and one from dad, in order for it to code in order to get cancer. Alright, and we'll learn more about dominant and recessive in the next chapter, but just know for recessive you have to have two copies, one from mom, one from dad, and with dominant genes you only need one copy. So tumor suppressor gene examples, P53. So 50% of tumors have mutated P53. That's a huge number. I feel like, and I'm sure it is, that this is a gene that should be extensively studied. If half of tumors are because this is mutated. And so what P53 does in its normal form, its non-mutated form, is it activates apoptosis if a cell is damaged. And a way that you can remember this is, I mean, this is kind of a stretch, but P53 apoptosis, it's got a lot of P's in there. So that's what activates apoptosis if a cell is damaged. So if it recognizes the cell is not normal or it's damaged in some way, P53 will come along and be like, sorry, bud, it's time to self-destruct, and apoptosis, programmed cell death. The BRCA1 is a major breast cancer gene. And this one should be easy to remember because I think this is, I'm not sure if this is why they named it that, but BR, breast, CA, cancer. And you can actually do a DNA test to tell you if you have these mutated copies. So I don't know if you guys are familiar with this story, but Angelina Jolie, the gorgeous actress, she had a history of breast cancer in her family, and so she got this DNA test and saw that she tested positive for having this cancer gene. And does that 100% mean she's going to get breast cancer? No, but is she highly susceptible to get it? Yes. And so what she did, and at the time, this was very, like, people were it was kind of a controversy. And in my head too, I was like, that's crazy. Why would she do that? But she had a complete mastectomy, so she had all of her breast tissue removed. Because if you don't have breast tissue, you can't get breast cancer. And so rather than taking that risk, she knew she was high risk for this type of cancer. She just got rid of the tissue before it could even turn to that. So now, knowing what I know, I understand why she would do that. But at the time, I want to say I was a teenager in my early 20s, I was like, I can't imagine doing that. I mean, but yeah. So, BRCA1 gene. And this codes for DNA repair enzyme. So again, these two are in their normal form. These are ones you should know. And what they code for. And then know what they do in their mutated form. So P53, if it's mutated, it's not going to activate apoptosis of these damaged cells. Okay, mutated P53 allows severely damaged cells to live. It's not going to activate that apoptosis of abnormal cells. And then mutated BRCA1 does not repair DNA damage. Sorry, my can's popping. Gotta have my Monster. So on to our question of the day. Will we ever prevent cancer from forming? That's a hard no. At least based on, I don't see how we ever could. Because what causes cancer are DNA mutations, which are random. And so we can't predict when and where DNA will mutate. And so there's just it's like predicting an earthquake. You might have an idea of where it could possibly occur or where is at higher risk, but you can never predict when and where that mutation will happen. So to summarize that, we have talked about DNA mutations. And how they cause cancer. And we cannot prevent nor predict DNA mutations. Are there things that you can control in your environment that can reduce the amount of carcinogens that you're exposed to and certain lifestyle things that can decrease your chances of getting these mutations? Yes, but ultimately we can't control or prevent DNA mutations in general. Okay, so oncology is the study and treatment of cancer. And tumors are classified based on their place of origin. So we're going to talk about some different types of cancers, and basically what you should know or remember from this is what it's called and where it's formed. So one type is carcinoma. And these are cancers of epithelial cells. And epithelial cells are cells that line things. Like they line the inside of your digestive tract. It's part of your epidermis. Sarcoma is cancer of the muscle, bone, or connective tissue. So if you know anything about physiology and anatomy of muscles, there are things called sarcomeres. So sarco is kind of referring to muscles. Leukemia, cancers of the blood. So in your blood you have leukocytes, which are white blood cells, if that helps you remember. Lymphoma is cancer of the lymphoid tissue, or tissues that aid in the immune system. And blastoma is cancer of an immature cell. So this is most often childhood cancer. So if you think about it, and I don't think there's a chart here, there's not. Typically we see a lot of cancers in older people. And so I'll call them seniors. So why would you see a higher incident of cancer in older people and seniors, what am I saying? People that have lived for a long time. The reason for that is they've had more time for these mutations to accumulate. Their cells have divided more times, allowing for more time for mutations to occur and accumulate and to result in cancers. Children typically get these cancers because they are inherited from their parents. So they're born with it, with the mutations, and eventually it can develop into childhood cancer. So, environmental cancers, 90 to 99% are caused by carcinogens. And this seems like a high number to me. It's probably actually a little lower. So seeing this brings a little bit of relief to me, because what that means that about 5 to 10% of cancers are inherited. So maybe I can chill a little bit about getting a brain tumor, even though it is in my genes. Most cancers are caused by things in the environment, carcinogens. That is a cancer-causing agent. And it acts by causing DNA mutations. So 3 examples of carcinogens are radiation. So this is used in x-rays. So when you go to the dentist and they put that heavy cape blanket thing on you, that's to try to block some of the radiation from getting to your body. This also comes from UV light, from the sun, from tanning beds. And then chemicals. I don't like the word chemicals because everything is chemicals, but carcinogenic chemicals, like tobacco smoke. We all know that smoking can increase the risk of cancers of the mouth, the larynx, the pharynx, the lung cancer, as well as other cancers too. And then asbestos. I'm sure you've heard the mesothelioma commercial if you've been exposed to asbestos commercials. And also, and I think this is very interesting, viruses can cause cancer. So there are 4 human viruses that are known to cause cancer. These are hepatitis B and C viruses, the herpes virus, and human papilloma virus. And like I said, I teach 7th graders, and my daughter also just went into 7th grade, and they recommend getting the HPV vaccine because it can prevent cervical cancer later on. And so I always hear my students talking about it, like, oh, I had to get a shot in the butt. It was my HPV shot. And then, yeah, my daughter was super nervous about it too. But the fact that we can prevent the spread of these viruses and therefore lower the incidences of these cancers, that's pretty amazing. And what these viruses do is it degrades p53 in infected cells. So remember, p53 activates apoptosis. And if it starts to degrade that protein to break it down in cells that are infected with this virus, then you can get cancer because the abnormal cells are told to self-destruct. Okay, this table here is not super inclusive of all cancers, but it shows side-by-side male and females. And, let's see. The number of deaths in thousands, and the main thing to note here is that lung cancer, compared to all these others like oral, larynx, esophageal, stomach, pancreatic, bladder, kidney, myeloid leukemia, cervical cancer, lung cancer is, in the number of deaths, is way, way up, and it's mostly attributed to smoking cigarettes. Now, something that I would be interested to know is about vaping. I know since I work with young kids, I know they don't typically go for smoking cigarettes. They're going to vape, and they do it often. I would be curious as to if that just falls into the chemicals category here or what. But I would be curious to know the numbers on that. And I think the problem is we don't have the numbers because it hasn't been a thing for long enough. But anyway, I do know among young people, the number that smoke actual cigarettes are down, way, way down. So anyway. So let's look at some treatments, cancer treatments. And the first one is surgery. So if it's possible to remove the cancer, cut out the tumor, they're going to do it. But then it's usually also coupled with one of the other 2 treatments we're going to talk about because you want to get that tumor shrunk and as small as possible before you remove it, and also to get any of cells that may have metastasized and moved to other parts of the body. So in my mom's case, and again, I don't want to sit and talk about my mom the whole time, but her brain tumor was in her medulla, so in her brainstem, and it was what's called diffuse. So it's not like it was a big chunky tumor. It was just those cancer cells were spread throughout her brainstem, and it was only an area the size of your pinky fingernail. It was tiny. But you can't cut it out, first of all, because that would be removing almost the whole medulla itself, which regulates blood pressure, heart rate, a whole slew of other things, speech, coordination of muscle movements, and yeah, my mom's case, surgery and removal of that wasn't possible. So they went to this next step called radiation. And this is for localized cancer. Like my mom's. It was just in one tiny spot in her brainstem. And so this is a picture of it, and it looks pretty scary, but basically this would be irradiating some sort of cancer in the head. And they wear these masks that hold them completely still, and then they have this really localized, I don't know if it's a beam or what, of X-rays or gamma rays. And what those do is they damage the cancer cell DNA. So you don't want to do this on cancer that has spread throughout the body, because then you're irradiating all the cells in the body, and then you can cause other mutations that can lead to other cancers. So you don't want to do that. So this is only for localized cancer, where they can target exactly where to send that radiation to try to damage the DNA in the cell. And chemotherapy. I'm sure we've all heard of this one. This is where you have body-wide metastasized cancer. So these are chemicals that interfere with cell division, DNA repair, and DNA replication. So it targets rapidly dividing cells, which are cancer cells. But there are also other cells in the body that divide rapidly, and it seems like I heard this somewhere. I need to double check, but cells like the lining of your epithelium of your digestive system are rapidly dividing cells, and that's why they can get like sores in their mouth and digestive issues and upset stomach when they're undergoing chemotherapy. And then I also want to say something to do with your hair follicles being rapidly dividing cells, and that's why they can lose their hair. I could be wrong. It makes sense to me, and that's what I've heard. And an example of this is Taxol. And we talked about this when we were talking about cells, because it inhibits spindle fibers. So the spindle fibers are what attach to the chromosomes to pull them apart during cell division. And if you can prevent them from attaching and being able to divide the genetic material of the cell, then the cells can't divide, and cancer can't do what it wants to do, which is divide uncontrollably. And the last one is immunotherapy. And this is some cool stuff. This is when they use your own immune system to fight the cancer. So on its own, your immune system is kind of crappy at fighting cancer, which is why people get it. But if you give it a little boost, then it's actually one of the best ways to fight it. So an example is the drug Herceptin. And then also what they can do is they can locate cells in your body that are actively fighting the cancer. They can remove one or two or however many of them and take them to the lab and replicate them and then put them back into your body. And so now you have this whole army of cells that know how to fight this cancer in your body, and that can help use your own immune system to fight it. And Herceptin blocks HER2 in breast cancer. All right, so I think that covered it all. And just a quick summary here. Sorry, I have my copies here with the answers. Know HER2 and what it does normally. Okay, so it codes for estrogen receptors. And then know what it does when it's mutated. So when it's mutated, it causes many of those receptors to be on the gene or on the cell. So estrogen can bind and stimulate mitosis. The RAS gene in its normal forms activates cyclins that help the cell through the cell cycle as needed, but when it's mutated, it just activates them all the time, so the cell divides and divides and divides. Tumor suppressor genes code for checkpoint proteins. And some examples are P53, which activates in its normal form apoptosis of cells, and so in its mutated form, it's not going to activate the apoptosis of abnormal cells, and so they can continue growing and dividing. And then BRCA1 codes for DNA repair enzymes in its normal form. And so when it's mutated, it's not going to repair DNA damage, and you can get these abnormal cells that keep dividing. And I wish I had the table to fill in with you, I'm sorry. But, like I said, that is a great way to review, is to print that off. If you can find it, I'll double check that it's on Canvas. If I ever say Schoology instead of Canvas, it's because that's the learning platform we use at my school. So if I ever say that, I'm sorry. But I would print off that table and go through that and see if you can fill that in. Use your notes to help you. You can possibly use flashcards with these genes and what they do. And yeah, I think that's it for today. So have a great day, guys.